Okay, so the title of the session today is How a Proteome Catches a Phenotype, which I absolutely love as a title. And also the title I was assigned is Losing Your Edge in Protein-Protein Interactions, which I like as well. I also was thinking yesterday about how to introduce this talk. You know I'm not an aging person, so I appreciate your invitation. I tried to change my research subject 6 months ago when you invited me, but it didn't really work. But I think we have common problems, and that's probably why you asked me to come here. And that commonality came up so beautifully in Michael Petracek's talk yesterday that I asked him if I could use this slide, and he graciously said, yes, thank you, Michael. And really, we're going to try to, I think it's the same for other people in this session, is to really go through this theme of aging as a deterioration of what I call interactome networks, but Michael said biological networks yesterday, it's the same thing. So you also mentioned proteoforms. This is something I'm extremely interested in. You probably know about this particular little perspective on trying to measure, and sort of write down a few things to try to get, the sort of estimated number of proteoforms out there. And of course, what's important to this meeting is how do these proteoforms age, but then more importantly for this session, when they age, if they age, what is then their new functions or loss of functions, interactions, etc. So I live more here on this side of things. I was used for many years to have nodes representing genes. And of course, we have to think it's just not good enough. We have to think of each one of these nodes as a set of proteoforms or a proteoform family coming out of this. And some of these representations here I could see are related to aging. But the talk is more on this side. How do we try to link interactome networks and complex traits? So the justification for a lot of what we do is really basically genetic. I mean, I grew up as a geneticist in Belgium, and I'm still a geneticist in heart. So at this point, 2020, 20 years after the human genome sequence, I think we have still a few problems that are interesting, actually, interesting challenges. But practically speaking, they should be solved soon. One of them is incomplete penetrance. How most genetic traits are actually really probabilistic. And you can have members of the same family with exactly the same genetic variant showing very different phenotypes. Some show the trait, others don't. One example that made the news, especially here in the States, but I think all over the place, was this amazing decision that someone like Angelina Jolie had to make based on the probabilistic idea that 87%, she had an 87% chance of developing breast cancer. To me, and I'm maybe perhaps a little Belgian on that side, it also means she had 13% chance of not developing breast cancer. And that's fascinating to me. And I'm wondering, what's behind that, right? And of course, this is true. This is true for most genes, most Mendelian traits, and most alleles in most genes of Mendelian traits. And the basic hypothesis here is that it's systems and networks in our cells and systems between cells, of course, at every scale you can imagine, that underlie genotype-phenotype relationships, right? Actually, one more example of that. Sorry, I should have put this slide first. But the other problem about after incomplete penetrance is really pleiotropy, right? How a variance in the same gene can lead to different phenotypes. How does that work, right? So incomplete penetrance, pleiotropy, go back to this concept of let's try to understand this relationship, genotype to phenotype, through perturbations of networks. So I'm suggesting to go through 3 vignettes in this talk. Again, since I'm not really in your field, I had one option, which was to talk about one thing. The other one was to sort of present a palette of different things. And that's what I decided to do. So we're going to go through a little bit of mapping tools and where the field is at. I think it relates to a lot of the stuff that aging is interested in. We'll go through very quickly our latest interactome reference map for human, published this year. And then we'll talk about Edgetics, which really is what Adam is waiting for, I think. All right, so tools. Here I have just 4 slides. It's one paper. It's an exercise to try to maximize binary interactome mapping with a minimal number of assays. I think what's interesting in biology is even in DNA sequencing, you really usually cannot map a huge object like the human genome with one method. You usually need a combination of different assays. This is certainly true for structural biology, for example, right? Where you need different assays, different versions of the assays to basically get to crystallize the proteins that you're interested in or the protein complexes. Binary protein-protein interaction assays are the same. I won't go through the details, but suffice it to say that most of them, Igor, I'm not sure if he's part of this meeting, Igor Stagliar, many others, Eric Vonker, there's a field of developing these tools. And what we did here is combine them. What they have in common is you split a given protein. Stanfield's had thought about the transcription factor, but you can have fluorescent proteins. You can reconstitute a pathway, a signaling pathway. You can even do biophysical interactions. And you can, the idea is to combine these assays the best way, but in an optimal manner. And also the versions of these assays, which you can see here, what we did is we split them into how you fuse your proteins, your pairs of proteins of interest, and where you express them. I'll just give you one example of what happened here. What this experiment is on the left side, 60 highly demonstrated protein-protein interactions, many of which are shown here. They haven't been all structurally solved, but it's very high quality in the literature. If I showed you examples, you'd go, okay. And as a negative control, we use 76, just that's the number, random protein pairs, which were randomly picked among all 20,000 by 20,000, which is 200 million combinations at one side, right? And that's the negative control. And that's one of the optimal combination that we came up with. A couple of assays on this side. You recognize some of them are new, sorry. And the beautiful thing is in each row are the protein-protein interactions we detect out of these previously demonstrated protein-protein interactions under condition for each row where none of the random protein pairs are detected. So we basically titrate everything such that in the good old way of biochemistry, basically, with your negative control, you titrate your conditions to maintain that at the highest level of specificity possible. You can see very quickly that with adding assays and versions thereof, you basically have this phenomenon where if you work with the right specificity in each case, the hope here is that we can get to really a viable set of assays and versions thereof, which we call the assay home, a little bit as a joke, to get to near full sensitivity of mapping of these binary interactions. So that's sort of, this paper is recent, and that's where at least we are, again, working together with a great number of labs on this one. Okay, second vignette is if we have good tools that people can trust, that we can use relatively at high throughput, we can hope to really get this landscape solved, right? But it's huge. We tried this 3 times so far. The latest, as I said, is this year in 2020. It's a reference map of the human binary protein interactome. And the 2 previous versions, the first one was in 2005, and the next one after that was 2014. So 3 attempts on our side. Now, there are, of course, other labs doing similar or related strategies to map. We're not the only ones, of course. It's just in 20 minutes, I can't go to what others have done. We can discuss that during the question session. But to me, these evolving maps, they feel a little bit like the early, historical geographical maps, 3rd century here, the Martinus map here in the 15th century. This is a beautiful one, the Waldsee-Muller one. And the point about this is it's 1, very hard. 2, when you look back, you go, oh my God, they weren't that great at generating these maps. But at that very time, with the technology you had, you did your best. And that's really where I think the philosophy of this is. Give you a sense of that. We're now, like I said, in this 3rd attempt, what we call the reference binary interactome map. We've screened the full space, really, of nearly 20. I mean, we always say 20K by 20K, but it's not exactly 20,000 genes in the human protein coding genes in the human genome. Also, some of them appear to be non-clonable. I mean, so this is basically, practically speaking, a full space, which we screened 9 times now. It was, half of the space 2 times in 2014, and that's the first effort here, over 10% of the search space. So Two little bits of information. Quality. Again, remember, I just told you about the tools, the binary interactome assayome type of idea. Just compare, if you would, purple to blue. Purple is the interactions that we've mapped, our map, relative to, and it's written over here, literature curated information with multiple evidence. So this is information provided by thousands of labs, curated by a few databases, multiple evidence, and we compare ourselves using either MAPIT or GPCA, in this particular case, two assays that I showed you previously in my assayome slide, relative to random pairs. And I hope you see that we are on par, if not in some ways, sometimes superior, but the details here don't matter very much. What might matter to you is the volume of information, and we need information to get this thing done. And this is in blue how in 2005, 14, and 19, the literature was at in terms of number of interactions available of high quality with multiple evidence. And this is the size of our map, about 4 times the size of what you have from literature curated information. Those are numbers, statistics, not rocket science, but perhaps this is more interesting to you guys. And that is, where do we find these protein-protein interactions in the space? So again, let me go through this very quickly. The search space is 20,000 by 20,000, right? 20,000, I summarize it always by 20,000 by 20,000. What we did here is we ranked the genes, the 20,000 genes, by the number of publications associated with each one of these genes. So, you know, P53 is here, and RB, and whatever. And then over here are the genes that were never studied by anyone. And then we rank them the same way in this orientation. And I hope you can see 2 things. Number one, we're evolving over time, we being our systematic map, but also the literature. But you can see how, I don't know for what reason exactly, what's always thought is sociological biases, but the field of basically molecular biology, really, of human biology, is heavily concentrated when it gets to finding contacts between gene products in a very small zone that we refer to as a den zone, while our map is more spread. Let me remind you, if I may, I'm sorry, that in terms of empirical proof of quality, that the purple is about equal, if not a little bit superior to blue. So again, you know, I hope you see that we're sort of going into unknown territory here. And this is particularly important if you want to do the next piece of the talk, which is coming in the next slide, which is using these reference maps to try to understand genetics. So here's what we did here is we don't take all 20,000 genes here, but on the left side, the Mendelian disease genes, which I'm going to be talking about in a minute, and also cancer genes. And you can see again that a good proportion of disease genes and cancer genes are in this zone where there's very little information yet in the literature, and it's catching up, but at its own pace. So the whole idea is and by the way, I'm not saying that purple here is complete. It's not complete. We don't know exactly how much of the interactome we have mapped so far, because it's still very hard to estimate the total number of biophysically relevant interactions out there. I can discuss that later. But it's a good chunk. Let me put it this way. All right, last bit, Edgetics. So this came up about a decade ago, and we started thinking of ways to try to interpret variants involved in human inherited disorders from an edgetic point of view, which we define as such. Some variants could, of course, lead to amorphs or null, of course, right? No problem, and we call that node removal. But to what extent would the landscape of human genetics be made of variants that affect specific edges or a small number of edges while leaving the other ones unperturbed? And to what extent could that explain different phenotypes coming from different mutations of different genes? So I'll show you 2 or 3 slides, I forgot on that, 2 I think. One is just very simple computational look, download variants from the human gene mutation database, HGMD, and just look at what the variants are likely to do. Are they likely to misfold or create an unstable protein, or a loss, or create a loss or gain? You're very interested in gain of interactions. I'll make a note of that in a second. Loss or gain of specific interactions, and more than half of previously identified sequence in people who have belong to families with a propensity to developing one or more of a human inherited disorder were of this type. The reason why this is potentially interesting is this, again, this is computational only, 11 years ago, is we then mapped where mutations were in different genes and looked at whether or not the location of the mutations relative to protein domains might have anything to do with the different syndromes that sometimes genes are involved in. So we looked at genes that coded for proteins with at least 2 domains involved in at least 2 diseases. We give you an example. PTPN11 here is known to be involved in 2 different syndromes, Noonan and Leopard. This is where you can map the mutations associated with the first syndrome, and this is where you can map the mutations associated with the other one. So you can start seeing what the theme is here, is if we could grab what sometimes is called the variome, right? I love this term. I didn't come up with it. It's basically all variants of all human genes, right? And compare the variants to a reference, which I don't want to call wild type or normal, just the reference, because at this point, what is wild type, what is normal? But at least let's take a reference. And that's what we did here in collaboration a few years later with Sue Lindquist and Mikko Taipale. This is a wonderful collaboration. You all know her, and it was just before she passed. It was wonderful collaboration. And what we did is to use 3 different approaches to characterize about 3,000 mutants in about 1,000 genes involved in at least 1 genetic disorder. We did it the Sue Lindquist way. We did it our way, protein-protein interaction profiling. And then we did it the Marianne Walhout way, which is for transcription factors, profiling the sites that they can bind to. And I don't have time to go through details, but you can see where this would go, right, relatively easily. We give you 2 pieces of data. One, this is just HSP90, but Mikko did a dozen chaperones. Only about 20–25% of alleles actually bound more to 1 or more chaperones. Example here is EGFR L858R, something like that. Point being, again, 75% of alleles involved in a human genetic disorder didn't seem to be affected from a stability or folding point of view. So what we did is profile them from the point of view of, again, protein-protein interactions. And again, the system is you have a number of partners for wild type, and then you look at all the mutants and you see how the profiles might differ. And this is the picture between all disease mutations that we were able to do in this analysis and common variants as a control, right? And you can see that most of the common variants are actually really behaving almost like wild type. And then we can see a defect for at least more than 30% of the disease mutations doing this. Now, remember, this doesn't mean that 43% of disease mutations are wild type. We call them pseudo wild type because we're not claiming that we're testing all functions, all interactions of all gene products. We're not. It's a profile that is representative of the status of the protein. And now experimentally, we can get into this idea of differentiating for a single gene, how a single gene like TPM3 can be involved in different syndromes by actually measuring the profiles of the corresponding alleles. And so the summary here, before I maybe have a few more slides, I don't know. How am I doing on time? I have no idea. Can someone tell me how many minutes I have? I'm sorry.
A few minutes left.
Few minutes, Adam? Okay, thank you. Sorry. So conclusion of what you saw here is we should maybe think of traits and as edgetic and consider this idea of edgetyping as a link between genotypes and phenotypes. And so if I have a few more minutes, I have 3 or 4 slides of what we're doing now, unpublished work, and it gives you, again, a sense, an example of where we are with variants of uncertain significance. This is the work by Anu Yadav, Georges Copin, and Florent Laval in our lab. And you can see how, from genome sequencing, how the number of VUSs has exploded. You all know this, but what do we do with this, right? So these are variants in genes known to be involved in 1 or more traits. We just simply don't know and what to tell people carrying these variants because we don't know what the gene product is doing with these variants. So we've pushed this idea now. Our goal in the next few years is to get to 100,000 variants clones such that we can express the corresponding proteins and compare them to their wild type counterpart. At this stage, we're about at 10% of that. We're close to 10,000 alleles. I don't mean to impress with numbers. That's not the idea. It's just that once you get into large numbers, you can start asking general questions, right? Of genetic architecture, landscape, etc. This is how some of these matrices look like, right? So in each little, I know we're at 50,000 feet here. You can't see it, but that's the point. Is each matrix is a gene with partners and mutants, and then we look at the relationship between these different things. And I can say two things. One is the observation from 2015 is confirmed. As a matter of fact, with deeper and wider edgetyping, we really see now the majority of pathogenic variants are edgetic. But more importantly, edgetic perturbation is directly proportional, it looks like, to clinical significance. So now we have pathogenic here and benign, and then the VUSs for which we don't know anything and how they alter the gene products. And you can see that they're somewhere right in the middle. Let me give you one example of that. This is MLH1. You don't need to look at the left, it's raw data. But then we do a semi-quantitative edgetic characterization on the right side. And I can't see, hold on, okay, here. This is the wild type MLH1 and its interaction profile, okay, about a dozen partners, something like that. And this is a number of alleles in MLH1 and how they can mediate or not some of these interactions with their profiles. And you can see, I'm sure right away where you are, that these things are going to cluster as shown. Why am I not doing this? Here. Oops, sorry. And no, so if you cluster alleles by alleles, you start seeing how alleles are behaving the same way relative to each other, relative to other alleles in the same gene. I'm not showing you, but you can, of course, use structural biology then to map where these differences are, and it's starting to really become fun. So the summary of this little part here of unpublished stuff is we think we're starting to really get a powerful tool to get to variants of unknown significance and differentiate them between those that look more like pathogenic, potentially, and those that would look more like benign. And the summary is here. I give you a sense of a little bit where we are with tools and how important it is to get, maximal specificity in a condition where you get good sensitivity. Our latest reference map is of good quality and covers a space that is greater than what is available from literature stuff. And it looks like one of the ways to start interpreting genetic traits is to treat them as edgetic, would be the summary of my talk. So this is my acknowledgement slide, and I'll take questions.